Meiosis is the reason behind genetic diversity, while mitosis is also important because unlike meiosis, this cellular process is present in all living organisms because the process of mitosis helps organisms grow and repair themselves. Now let's discuss the meaning of these two and its process. Mitosis, it says here that it is the cell's copied genetic material separates and the cell prepares to split into two cells. Meaning, it is a process of cell duplication or reproduction. Strictly applied, the term mitosis is used to describe the duplication and distribution of chromosomes, the structures that carry the genetic information. This allows the cell genetic material to pass into the new cells. It says here that the resulting daughter cells are genetically identical. Where do I find the DNA? Chromosomes are in the nucleus of every cell. Chromosomes are made up of DNA. And genes are pieces of DNA that contain the instructions for building a protein. That's DNA. So we have here the four stages of mitosis. The first one is the prophase, metaphase, anaphase, and lastly telophase. Let us remember the word PMAT so that we can always remember the four stages of mitosis. So the first stage is the prophase. It says here that in prophase, the nucleus disappears. The spindle fibers form in the cytoplasm. The spindle fibers attach to sister chromatids. When we say prophase, it is the starting stage of cell division in eukaryotes. In both mitosis and meiosis, prophase is recognized by the condensing of chromosomes and separation of the centrioles in the centrosome. So this organelle controls the microtubules in the cell, and each centriole is one half of the organelle. During prophase, they separate to provide microtubule centers in each new cell. So we have here the example of prophase. The next one is the metaphase. The sister chromatids are pulled to the center of the cell, and it also says here that they line up in the middle of the cell. Metaphase is a stage in eukaryotic cell division in which the chromosomes align in the metaphase plate, as what I've said earlier. It is in the middle of the cell. The cell performs a series of checkpoints to ensure that the spindle has formed. The microtubules emanating from each side of the cell attach to each chromosome. As the microtubules are retracted, an equal tension is applied from each side of the cell, the chromosomes. This moves them to the middle of the cell. So after the metaphase, the sister chromatids that comprise the chromosomes are divided. At yung process niya is the process of cell division is completed. The next one is the anaphase, which is the third stage of mitosis. It says here that spindle fibers begin to shorten. The sister chromatids are already pulled na siya to the opposite ends of the cell. As you can see in the picture, anaphase, in which the chromosomes are segregated to opposite poles of the cell. It starts after the cell passes the spindle formation checkpoint, which allows chromosomes or chromatids to separate. Lastly, telophase. Of course, it is the final stage in cell division. During telophase, the nuclear envelopes are formed around the new nuclei in each half of the dividing cell. The nucleolus or ribosome-producing portions of the nucleus return. As the cell has finished moving the chromosomes, the main parts of the spindle apparatus fall depolymerize or fall apart. As telophase moves toward completion, the chromosomes release from their tightly bound structure back into loose chromatin. Telophase is ended by a process known as cytokinesis, which leaves the cell into new cells. So now let's discuss or define what is cytokinesis. So we have here its image. Cytokinesis is the division of the cytoplasm. It results into two separate total cells with identical nuclei. In biology, the process by which one cell physically divides into two cells, that's cytokinesis. It represents the major reproductive procedure of unicellular organisms, and it occurs in the process of embryonic development and tissue growth and repair of higher plants and animals. So we have here the example in cytokinesis in animal cells and in plants. In animal cells, it is accomplished by using microfilaments to pinch the cytoplasm. We have here the picture. And sa baba naman, in plants, a cell plate forms between the two daughter nuclei. So we're done in mitosis. Here is the picture of our real-life cells dividing. Now let's go to the meiosis. Meiosis, also called reduction division of a germ cell involving two fissions of the nucleus and giving rise to four gametes or sex cells, each possessing half the number of chromosomes of the original cell. So the meaning of meiosis here is it is the process of cell division that produces haploid gametes. The half, the number of chromosomes, we have here 23. So here is the example of meiosis. Now we go to reduction division. Meiosis in the process of cell division that produces gametes with the half number of chromosomes as somatic cells. So we have here cell undergoes two rounds of cell division. Since the sperm and the egg contain only half the number of chromosomes, they cannot be formed from mitosis. So that's why reduction division is in the meiosis. Did you know that humans have 46 chromosomes in their somatic cells? That's a trivia. Next one. We have here the picture of a sexual life cycle. Unique features of meiosis. Feature 1, synapsis. It says here that following chromosome replication, the homologous chromosomes pair all along their length. This process is called synapsis. Let us not forget the word synapsis and its meaning. The feature number 2 is the crossing over. While the homologous chromosomes are joined, crossing over occurs. Crossing over meaning it is the exchange of genetic material from homologous chromosomes. This causes genetic variations. So this is the picture of synapsis and crossing over. The feature number 3 is the reduction division. As what we've tackled earlier, the chromosomes are not copied in between the two divisions. At the end of meiosis, each cell contains 1/2 the genetic material, haploid or N. So here's the image of reduction division. Now let's go to the meiosis 1. Single centrosome replicates, so it forms two centrosomes, preceded by interphase. It says here, chromosomes are replicate to form sister chromatids. When we say sister chromatids, it is genetically identical and joined at centromere. So here's the picture of meiosis 1, the process, chromosome duplication, sister chromatids, and chromosome distribution to daughter cells. We have here the prophase 1. Individual chromosomes first become visible. So, na-define na natin yung crossing over. In prophase 1, homologous chromosomes become closely associated in synapses. So, crossing over occurs here. next one is metaphase 1. The homologous chromosomes line up in the center of the cells and are still held together, as you can see in the picture. Now let's go to the anaphase 1. Spindle fibers shorten, the homologous chromosomes are separated, the sister chromatids are still paired. When we say independent assortment, it is a random chromosomes moved to each pole. Some may be maternal and some may be paternal. That's independent assortment. Now let's go to the telophase 1. The nuclear membrane forms around each daughter nucleus. Each new cell contains two sister chromatids that are not identical due to crossing over. The nuclear membrane reforms around each daughter nucleus. So at the end of the meiosis 1, you have made two cells. Each cell contains a haploid number of chromosomes, so one copy of each chromosome. So in humans, each haploid cell has 23 chromosomes. No DNA replication occurs between meiosis 1. Let us not forget that. So now let's go to the prophase 2. Nuclear membrane breaks down again in prophase 2. In metaphase 2, the chromosomes line up in the middle of the cell. In anaphase 2, the spindle fibers shorten and the sister chromatids move to opposite poles, as you can see in the picture. Telophase 2, nuclear envelope reforms around the four sets of daughter chromosomes. It's 4 haploid daughter cells. So at the end of the meiosis 2, there are 4 haploid cells, meaning only one copy of each chromosome. So, as what I've said earlier, for humans, each haploid cell has 23 chromosomes. Now, no two of these haploid cells are alike due to crossing over. Earlier, I already said that at the end of meiosis, no DNA replication occurs between meiosis 2. And I repeat that in meiosis 2, no two of these haploid cells are alike due to crossing over. This is why you and your siblings are genetically unique. Yes, we are all unique.